morning, ladies and gentlemen. Thank you for joining me today for my presentation about microRNAs, their role in COVID disease, and our unique method used for their quantification. Unfortunately, I assume that there is no need to introduce the disease, so-called COVID-19. It's an illness caused by coronavirus SARS-CoV-19, which has brought a pandemic outbreak ever since the beginning of 2020. Due to quick spreading and variability of severity of this disease, there has been a massive development of diagnostic tools to detect the virus presence in a human body. Two basic sample specimens are used standardly for patient's examination. First of all, it's swab, all kinds of them, either from nasopharyngeal region, as those are the most accurate ones, throughout nasal swabs and oral swabs. These are usually subsequently used for nucleic acid isolation, followed by reverse transcription PCR or digital droplet PCR or LAMP tests. Another example of swabs usage is POC testing, commonly used for antigen detection. All of these approaches provide us with one and only information, and that's whether or not the person is positive for SARS-CoV infection. Testing can be supplemented with a blood sample analysis, using usually for the presence of the antibodies, which shall conclude about being presently infectious or previously infected by COVID virus. But the question is whether these information are truly sufficient. Would it be possible to perform a test which would give us much more information about the infection and the patient himself? BioVendor is a biotechnological company interested in microRNAs and their quantification for a while. So once the COVID-19 started to spread around the world, our microRNA team has been wondering how to use our knowledge and tools to help with stopping the global pandemic. The solution came in several original publications dealing with virus SARS-CoV-19 and its microRNAs. These primary research articles used either in silico modeling or next-generation sequencing data to predict putative regulatory sequences known as microRNAs, theoretically produced by COVID itself. Based on these, our team of specialists have developed unique assays to detect these viral molecules from nasopharyngeal swabs. Also, we have designed several assays for full panel of human targets detected in serum involved in immune response to viral infection or risk of development of various secondary complications or organ regulation in proper functioning. So our tool should provide you with much more information, not only whether the patient is positive, but also whether the patient is at risk of severe course of the disease, what secondary complications could develop, and which organs are at high risk of temporary or even permanent damage. I realize that today you've already heard a lot about the microRNAs, but Please let me just briefly describe and characterize these short non-coding regulatory sequences. I believe that all of you know the central molecular dogma, where genomic DNA is transcribed into messenger RNA, which is subsequently translated into a protein. This was pretty much universal model for all eukaryotic cells, but it has become slightly more complex and complicated once the purpose of non-coding RNA sequences was described. Long non-coding RNAs play a crucial role in condensation of genomic DNA. So they are involved in heterochromatin and euchromatin formation. These long molecules also regulate the expression of the second group of non-coding RNAs, small and mid-sized non-coding RNA. They are usually playing an important role in post-transcriptional or post-translational regulation of gene expression. These short and mid-sized RNAs are the main targets of biovendor assays, particularly microRNAs, PV-interacting RNAs, and small nuclear RNAs. So, microRNAs are the products of genomic DNA transcription. In this phase, they are called primary microRNAs, and they have double-stranded structure with stem and loop organization. These primary microRNAs are usually processed by Drosha complex into precursor microRNA. This is transported outside the nucleus into the cytoplasm. Precursor microRNA is spliced and further unwinded by dicer and other mechanisms into mature microRNA. This mature sequence is presented in all its precursors. Once the maturation is finished, the microRNA plays its role either by transcriptional repression, where it binds the complementary messenger RNA and thus blocks its translation, or by messenger RNA destabilization followed by mRNA degradation. This way, microRNAs regulate and control all the molecular signaling pathways and all the possible levels. Therefore, microRNAs worth the attention of each and every molecular biologist. There is a reason why microRNAs shall be recognized also by all scientists and mostly clinicians. MicroRNAs are very stable and well detectable in all biofluids. Once the cell produces these non-coding molecules, it can either use it for its own regulations, or the cell exports microRNAs into the system. MicroRNAs could be packed into various microvesicles, exosomes, or apoptotic bodies. They can be carried by high-density lipoproteins or RNA-binding proteins. Their destinations are recipient cells, which processes their message and responds accordingly. Due to these shuttling processes, microRNAs could be detected in all biofluids, including blood, urine, synovial fluid, cerebrospinal fluid, tears, sweat, breast milk, or feces. Now, when you all are hopefully interested in microRNAs and other non-coding RNA molecules, it's time to discuss their quantification, which honestly used to be not easy at all. MicroRNAs are very short. They usually consist of only 19 to 23 nucleotides, so the standard primer design wouldn't be effective. Mature microRNAs completely lack any common sequences like poly-A tail or the cap. MicroRNAs are also rich in GC content, what usually causes increase in melting temperature. They are so-called microRNA families in which members differ barely in one or two nucleotides. MicroRNAs could have various biochemical modifications on their terminal regions. And do not forget that the mature microRNA sequence is present in each of its own precursors. Therefore, you need to make sure that the assay is clearly recognizing and measuring only this mature variant of the target molecule. The two-tailed PCR has been already perfectly described to you by its own inventor, Dr. Kubista. Therefore, I would just quickly remind you that this assay, unlike the majority of our competitors, uses only target-specific two-tailed reverse transcription primer. So it creates very specific cDNA library which enters the PCR process. Of course, we use only microRNA-specific PCR primers for quantification and amplification in the polymerase chain reaction. Therefore, the two-tailed PCR reaction has superior specificity and incredible sensitivity, which are together so unique on the microRNA quantification market. As biovendor, and therefore also our team of specialists and scientists, have this amazing tool available, we searched for new publications which would be dealing with SARS-CoV-19 and its microRNA molecules. When we look at the COVID genome, there is the spike protein gene, S gene, which is the main focus of the most COVID-19 vaccines. But there are also other interesting regions. There is a non-structural protein 1 region, NSP1, which is also referred to as the host shutoff factor, where SARS-CoV microRNAs are encoded as well. There is also another microRNA encoding region within the M gene or the membrane glycoprotein gene. These various microRNAs have distinct roles. They modulate the host immune system at multiple levels, like chemokine signaling or inflammatory response. They also target the virus to different organs, like lungs, spleen, or gut. Viral microRNAs regulate cellular metabolism, cell cycle, apoptosis, and they manipulate the host mechanisms to replicate the viral genome. But the human body uses its own microRNAs to fight back. Not only that directly infected cells produce microRNAs to fight the inflammation and spread the information, also other involved tissues adjust their microRNA expression profile. We already know many microRNAs which could predict various conditions like thrombosis or cardiovascular diseases. Therefore, we put together a pre-selected panel of 20 different human microRNAs and 7 viral microRNAs to search for the most accurate and related to the COVID-19 disease and development targets. Based on their molecular function, we could cluster our targets into specific groups. There are microRNAs related to the cardiovascular risk, thrombosis risk, and lung invasion risk. The rest of the microRNAs have a major role in immune system orchestration and response to the infection. Our R&D team, as the first one in the history, has developed several quantification assays specific for SARS-CoV-19 microRNAs. Not only that we successfully developed them, we also succeeded with the real microRNA measurements from the patient's swab samples. So eventually, we verified previously mentioned studies in vivo and confirmed the presence of viral microRNAs. At this moment, we are at the very starting line and we measured only a limited number of samples. So no final conclusions can be made. On the other hand, we could already identify COVID microRNAs which cluster all samples together, and those which targets separate our patients into different groups. For example, we could identify samples 1, 4, and 9 to stay in a subset, with sample 9 having the highest, sample 1 having the average, and sample 4 the lowest microRNA expression. This trend was observed for 5 different targets. All these 3 patients suffered with bilateral pneumonia. We also used patient serum to quantify and profile several human microRNAs. Very interesting result came from the measurement of miR214 for a patient with sclerosis multiplex. This condition is known to have increased expression of miR214 due to its involvement in T helper cells differentiation. But this patient sample revealed increased expression in other microRNA targets as well. This observation was common also for other patients, number 6, 8, and 1. Very specific example was noticed for patient number 3 with elevated miR124. This patient suffered with thrombosis, and miR124 is known to play a crucial role in coagulation pathway and thus in thrombosis risk. Our aim is to develop the complex diagnostic tool with reasonable and manageable number of measured microRNA targets. Once we identify correlating microRNAs, we plan to develop multiplex reverse transcription reaction to minimize the price and wet laboratory work. We plan to prepare sample evaluation algorithm or program, which would process sample results and summarize them to simple report. At this moment, we are negotiating the cooperation with the largest hospitals to provide us with the sample and clinical data of patients to run our trials with conclusive number of samples. Also, it's already possible to purchase our assays for your own projects and use them for your novel studies. So at this moment, I would like to thank you so much for your attention and open a discussion for your questions.